Welcome back to the Deep Dive. This week we're doing a deep dive into the latest longevity research, focusing on papers published this week from March 10th to the 13th on longevitypapers.com.
Yeah, and we'll be zeroing in on the research that's most likely to make a splash in the biotech world. You know, the really surprising findings from credible institutions, the kind of stuff that might even make us rethink our current aging models.
Exactly. I mean, imagine the implications of these new findings, for researchers developing new therapies and interventions. There's a lot to unpack here, so let's jump right in.
Okay, sounds good. Let's kick things off with a paper that just came out on March 12th on bioRxiv. It's titled TL Path Predicts Telomere Length in Human Tissues from Histopathology Images from the Sanford Burnham Prebys Medical Discovery Institute.
Okay, TL Path, tell me more about this one. It really caught my attention. Predicting telomere length just by analyzing tissue architecture from standard histopathology images. That's really something.
It is. What's really remarkable is that they've been able to train a deep learning framework to actually detect age-related architectural changes in tissues that correlate with telomere length. And get this, it can do it without even knowing the age of the sample.
So it's not just matching chronological age with telomere length. It's finding actual structural markers within the tissue itself that can tell us something about biological age.
Exactly. They tested this on various tissues and found that they could predict bulk tissue telomere length with a pretty high correlation, actually outperforming chronological age as a predictor.
Yeah, I remember reading about that. The correlation for TL path was over 0.51 in well-represented tissues, while chronological age only had a correlation of 0.2. That's a pretty big difference. But what really intrigued me was the idea of age-discordant telomeres.
Right. Telepath was able to flag cases where individuals had shorter telomeres than expected for their age, or the opposite, longer telomeres than expected. Think about what that could mean for personalized medicine.
I mean, imagine being able to assess an individual's biological age and disease risk based on their telomere length just by using routine biopsies. It could revolutionize preventative health care.
Absolutely. And it could also be a huge game changer for researchers studying aging and age-related diseases. All of a sudden, they've got a tool to identify individuals with accelerated or slowed aging at the cellular level, even if it doesn't match up with their chronological age.
It opens up a whole new way of understanding the factors that influence aging and developing targeted interventions.
Yeah.
I really feel like we're on the verge of a new era in longevity research.
I agree. And speaking of new tools for assessing biological age, there's another study published this week on March 7th in Journal Science that's worth discussing. It's titled, Various Diseases and Conditions Are Strongly Associated with the Next Generation Epigenetic Aging Clock, Cheek Age.
Cheek age, I remember seeing that one. What really stood out to me was the fact that they can measure it from a simple cheek swab. I mean, that makes it incredibly accessible for large-scale studies and clinical applications.
Absolutely. And the team from Tally Health found that cheek age actually outperforms other epigenetic clocks in its association with a whole bunch of different health variables. We're talking about like 33 different diseases and conditions.
33, okay, that's a lot. What kind of conditions are we talking about?
Well, some are expected, like diabetes, liver disease, but what's really interesting is that this cheek swab test seems to correlate with things you wouldn't necessarily expect, like exposure to environmental chemicals or even someone's risk for depression. That suggests it's really tapping into something fundamental about biological age.
So it's not just predicting chronological age, it's actually reflecting the underlying biological processes that contribute to aging and disease.
Exactly. Imagine a future where a simple cheek swab could give you insights into your risk for a whole range of conditions. I mean, that could totally transform preventative health care.
Okay, let's move on to a paper that was published in Aging Cell on March 6th. This one really digs into the molecular mechanisms of aging. It's called Telomere Position Effect Over Long Distances, acts as a genome-wide epigenetic regulator through a common ALU element.
Ah, yes. This study from Aix-Marseille University is a really interesting one. It actually challenges our understanding of how telomeres influence gene expression and potentially, the aging process itself. They found that telomeres are acting as genome-wide regulators through something called TPE-OLD.
TPE-OLD, that's telomere position effect over long distances, right?
Mm-hmm.
I remember reading something about it involving these repetitive DNA sequences called ALU elements. I always thought that was just junk DNA.
Yeah, that's right. And for a long time, ALU elements, which are scattered throughout our genome, were considered non-functional. But this study suggests that they might actually play a much bigger role than we thought, especially when it comes to aging.
So how do these ALU elements in TPE-ALD actually work? Can you break it down for me a little bit?
Sure. So essentially, they act like switches that can turn gene activity up or down, and their activity is influenced by telomere length. It all comes down to how DNA folds and interacts in three-dimensional space. Imagine it like this. As telomeres shorten with age, they change the spatial organization of the DNA, right? And this affects which genes are accessible and how they are expressed.
So it's kind of like a domino effect.
Huh.
Telomeres shorten, DNA folds differently, and suddenly gene activity is altered all over the genome.
Exactly. And to make things even more interesting, they discovered a specific protein, RBPJ, that acts as a bridge between these ALU elements and the telomeres.
So there's a specific molecular mechanism that we could potentially target. If
Hmm.
we could somehow modulate this RBPJ protein or prevent those ALU elements from being influenced by the shortened telomeres, could we slow down the aging process?
That's the million-dollar question. It's definitely too early to say for sure, but this study definitely opens up a new avenue for research into potential interventions. It's really a fundamental shift in how we understand the role of telomeres in aging.
It definitely makes those ALU elements, that so-called junk DNA, a lot more interesting. Okay, speaking of interesting, let's move on to a paper that has people buzzing about a potential new way to target senescent cells, those zombie cells that accumulate as we age. This one's titled Cell Surface ARMP1 is a senescence marker in aging and idiopathic pulmonary fibrosis and was published on bioRxiv on March 6th by researchers at the Lifespan Research Institute.
Yeah, this one's really interesting. They've identified a new biomarker for senescent cells called LMP1, lysosomal associated membrane protein 1, and the key here is that it's found specifically on the surface of these cells.
Ah, I see. So instead of trying to target them based on their internal workings, which could be really tricky, we could potentially go after this external marker.
Exactly. It's like having a homing beacon for therapies. And they validated this in both human and mouse cells, showing increased LMP1 positive cells in mice with lung fibrosis, which is a serious age-related disease. But what's really promising is that they tested a dual antibody drug conjugate strategy to eliminate these LMP1 positive cells, and it seems to be highly targeted.
So it's like a smart bomb approach, taking out senescent cells while leaving the healthy cells alone.
Exactly. It's an exciting step towards developing effective senolytic therapies that could be huge for treating a range of age-related diseases. Imagine a world where we could clean up these harmful cells and rejuvenate tissues. It could change the game for conditions like lung fibrosis, heart disease, and even Alzheimer's.
It's definitely promising. And while it's still early stage research, it's definitely an avenue that deserves a lot more exploration. Now, speaking of promising avenues, let's shift gears and talk about a couple of plant extracts that have shown some really interesting potential for slowing down biological aging. The first one is from a paper titled Unveiling the Geroprotective Potential of Monarda didama L, which was published in Geroscience on March 11th.
Ah, Monarda didima, also known as bee balm. This study from the University of Padua is really interesting because they went beyond just lab testing and actually conducted a human clinical trial.
Bee balm, isn't that the plant that attracts hummingbirds? I'm curious, what did they find in the lab? And more importantly, what were the results of the human trial?
Well, in the lab, they found that an extract from Monarda didima showed some really strong antioxidant activity, reducing telomere shortening and protecting against DNA damage. But in the human trial, they observed some really remarkable results.
Okay, I'm all ears. What were these remarkable results?
So the participants who were taking the extract actually showed improvement in their leukocyte telomere length, and their DNA methylation age, which is a measure of biological age, actually stabilized. But here's the kicker. The placebo group actually showed signs of accelerated epigenetic aging.
So while one group was biologically aging faster, the group taking the bee balm extract seemed to be slowing down their internal clock. That's really impressive. Were there any other noticeable benefits?
Yes. The participants who took the extract also reported a better quality of life. They specifically mentioned improvements in their physical well-being, including things like movement and sleep quality. It seems like this plant extract might actually be a natural way to promote healthy aging.
That is really fascinating. It makes you wonder what other natural compounds might be out there just waiting to be discovered, things that have the potential to combat aging.
Absolutely. And while we're talking about plant extracts with anti-aging potential, another paper published on March 11th, this one in the journal Rosa, really caught my eye. It focuses on the protective effects of Rosa Ruxburgii trat extract, or chestnut rose, against UVB-induced inflammaging.
Chestnut rose extract. Okay, that sounds interesting. But hold on a second. Can you back up a bit and tell me what exactly is this inflammaging you mentioned?
Sure. So inflammaging is basically chronic low-grade inflammation that happens as we age. It contributes to a whole range of age-related diseases. This study, which comes from the Yunnan Botany Biotechnology Group, found that chestnut rose extract can protect against the type of inflammation that's triggered by UV exposure, which, as you know, is a major contributor to skin aging.
Ah, okay. So it's not just about preventing wrinkles and age spots. It's about actually targeting that underlying inflammatory process that drives skin aging.
Exactly, and they actually discovered how it works at the cellular level. They found that the extract reduced the production of inflammatory molecules in skin cells that were exposed to UVB radiation, specifically by blocking the activation of the IL-17 pathway.
So it's a targeted approach, going after a specific pathway that's involved in inflammation. That's pretty exciting. Did they find any other effects on the skin cells?
Yes. They found that the extract also protected against some of the damaging effects of inflammation on skin cells, things like reducing mitochondrial dysfunction and the production of those senescence-associated secretory phenotype factors, or SEA factors as they're known.
Ah, yes. SASP factors. Those are the troublemakers that can spread inflammation to neighboring cells, accelerating the aging process.
Exactly. So this extract could potentially stop inflammation in its tracks, before it can really wreak havoc and cause further damage. This could lead to new skincare products that effectively protect against sun damage and premature aging. But it also makes you wonder, could targeting specific inflammatory pathways be the key to not just preventing skin aging, but also preventing other age-related diseases as well?
That is a really interesting question. We know that inflammation plays a key role in a lot of age-related diseases. So if we could find ways to control it, could we potentially extend both lifespan and health span?
Absolutely. It's a central theme in longevity research, and one that we'll continue to explore as we dive into some of the more novel models and insights that are coming out of the latest studies.
Model organisms, here we come. I'm always fascinated by the creative ways researchers study aging. What have you uncovered in this week's collection of papers?
Well, there's a paper on bioRxiv from Duke University that was published on March 6th that describes a really innovative artery-on-a-chip model that they're using to study arterial health. And the cool thing is they're using human stem cells for this.
Artery on a chip. Okay, that sounds super cool. So they've basically created a miniature human artery in a lab.
Essentially, yes. They used human-induced pluripotent stem cells, or iPSCs, to create a miniature functional artery that actually mimics the structure and function of a human artery.
But how does this tie into longevity research? What can we learn from this model that we couldn't learn from, studying arteries in a living organism?
Well, one of the hallmarks of aging is the stiffening of our arteries. That makes them less efficient at delivering blood and oxygen to our tissues.
Ah.
And this is a major contributor to cardiovascular disease, which, as you know, is a leading cause of death worldwide.
Right, so by studying this artery on a chip model, researchers can get a much deeper understanding of how aging affects arterial health at a cellular level, potentially leading to new ways to prevent or even treat cardiovascular disease.
Precisely. And they've actually already shown that the model can mimic disease states. They were able to model polycythemia and Hutchinson-Gilford progeria syndrome, which is a rare genetic disorder that causes accelerated aging.
So they can actually observe the effects of these diseases on the artery in real time, getting those insights into the underlying mechanisms and potential therapeutic targets.
Exactly. It's incredible. And because it uses human cells, it's much more relevant to human health than traditional animal models. Plus, it opens up possibilities for personalized medicine.
Imagine a future where we can test the effectiveness of drugs on your specific cells before prescribing them. That would be amazing.
Right. It's a powerful vision, and this artery-on-a-chip model is a big step towards that.
Now, we can't talk about model organisms without mentioning zebrafish. They're a favorite for studying aging, and there's a paper on bioRxiv from MDIBL, published on March 7th, that introduces a new zebrafish model. This one could shed light on sarcopenia, the age-related muscle loss that affects so many people as they age.
Oh yeah, the atrophish, as they call it. They've engineered the zebrafish model to have increased expression of atrogen-1, which is a protein that's linked to muscle atrophy. They're basically speeding up the aging process in the muscles of these fish.
So they're creating a fast-forward version of muscle aging in the zebrafish. That's really interesting. What kind of observations have they made so far?
Well, they've observed muscle atrophy and impaired movement in both the larva and the adult fish, which was expected. But what's really interesting is that they found some evidence suggesting that muscle atrophy might actually contribute to neurodegeneration in the central nervous system.
Wait a minute, hold on. Are you saying that muscle loss in the body could be triggering changes in the brain?
It's a really surprising finding, and it definitely needs further investigation, but it points to a potential link between muscle health and cognitive function as we age. They also found that the adult atrophish actually had impaired muscle regeneration, which is really similar to what we see in aging mammals.
So this atrophish model could be really valuable for studying not just the mechanisms of sarcopenia, but also that complex interplay between muscle aging and neurodegeneration. It really highlights how interconnected different systems in the body are, especially as we age.
Exactly. And if we can figure out how to prevent or reverse muscle loss, could we extend both lifespan and health span? It's a really interesting question that the atrophish might help us answer.
Now, for our final paper in this section, let's talk about mitochondria, the powerhouses of our cells. There's a study published in Nature Aging on March 7th that reveals a really surprising role for mitochondria in aging hematopoietic stem cells, the cells that give rise to all of our blood cells.
Ah, yes. This study from the Cancer Science Institute of Singapore is a fascinating one. It's titled Mitochondria-Enriched Hematopoietic Stem Cells Exhibit Elevated Self-Renewal Capabilities, Thriving Within the Context of Aged Bone Marrow. It really challenges the traditional view of how mitochondria function in aging stem cells.
I remember learning that mitochondria tend to become dysfunctional and accumulate damage as we age. But this study seems to be saying something totally different.
Yeah, it does. They actually found that hematopoietic stem cells, or HSCs, with high mitochondrial mass in aged bone marrow actually have enhanced self-renewal capabilities.
Wait, so you're saying that they're not just worn-out cells with damaged mitochondria, but they're actually more robust and have a higher capacity to replenish themselves? That seems kind of counterintuitive.
It does, but it suggests there's more to the story than just simple mitochondrial decline in aging stem cells. And get this, they identified a specific marker, GPR 183, that distinguishes these unique aged HSCs, the ones that have that high mitochondrial mass and a high capacity for self-renewal. It's like these HSCs have adapted to thrive in the aged bone marrow environment.
So they're not just surviving, but they're actually thriving in this more challenging environment. It's really interesting. Could manipulating mitochondrial function in stem cells be a way to rejuvenate the blood system during aging?
It's a fascinating question, and it opens up a whole new avenue of research. I mean, imagine if we could harness the power of these supercharged stem cells to combat age-related blood disorders and even improve immune function.
It's amazing to see how much we're learning about the complexities of aging and how researchers are developing new tools and therapies with the potential to really transform how we age.
Yeah.
It's an exciting time to be following this field.
It really is. And this is just the beginning. Stay tuned for much more to come. So we've seen some really cool stuff, how researchers are using these cutting edge tools like deep learning and organ on a chip models to really get into the nitty gritty of aging. And then we've also seen some really cool potential of, natural compounds and targeted therapies to slow down or even reverse aging. But the quest to understand and extend human health span, I mean, that goes way beyond just these areas.
You're so right. I mean, there's a whole world of research out there exploring, different aspects of aging from the brain to the immune system and beyond. What else have you come across in this week's collection of papers? What really stood out to you?
But one paper that really caught my eye was published on bioRxiv just this week, March 7th. It's from the Institute for Regenerative Medicine at Texas A&M University, and it focuses on a potential new therapy for age-related cognitive decline.
Oh, cognitive decline. That's something that hits close to home for a lot of people. What's this new therapy all about?
So the title of the paper is Intranasal Human NSC-Derived EVs Therapy in Late Middle Age Can Restrain the Activation of NLRP3 Inflammasome and CGS-STING Signaling in the Age Hippocampus.
Okay, I'm going to be honest with you. That title is, it's a bit of a mouthful. Can you break it down for me? Like, what are these EVs and why are they being delivered intranasally?
Sure. So EVs stand for extracellular vesicles. They're these tiny packages that are released by cells, and they contain all sorts of molecules, including proteins and microRNAs. And intranasal delivery, that just means they're being administered through the nose.
Okay, so instead of injecting these EVs into the bloodstream, they're delivering them directly to the brain, like through the nose. Is that to get around that blood-brain barrier, that protective shield around the brain.
You got it. The blood-brain barrier can make it really tricky to deliver therapies to the brain, so this intranasal delivery method is pretty innovative. And in this study, they used EVs that were derived from human neural stem cells, or NSCs.
Wow, so they're taking these tiny packages from neural stem cells and delivering them right to the brain. That's pretty cool. What's the idea there? Like, what's the hope with using these NSC-derived EVs?
Well, the hope is that these EVs can actually rejuvenate the aging brain, by delivering these beneficial molecules that can counteract the negative effects of aging on brain cells.
Hmm.
And in their study, they actually focused specifically on the hippocampus, which is a region of the brain that's crucial for memory, and it's particularly vulnerable to age-related decline.
Makes sense. So they tested this out in mice, right? Did they find that these EVs actually had any kind of impact on cognitive function? Did they work?
Yeah, they did. They actually found that when they administered these EVs intranasally in late middle age, it actually led to a reduction in oxidative stress and neuroinflammation in the hippocampus.
Ah, oxidative stress and inflammation are usual suspects when it comes to aging, right? But it's really interesting that they were able to target those processes specifically in the brain. And did that actually translate into any improvement in cognitive function in these mice?
Yes, the mice that received the EV treatment actually performed significantly better on memory tests than the mice that didn't.
Wow, that is really promising. It suggests that this approach could potentially be used to prevent or even reverse age-related cognitive decline in humans, right? But
Yeah.
of course, it's still early research.
Absolutely. We need more research to see if these findings actually translate to humans, but it's definitely exciting to see these kinds of innovative therapies being developed.
Yeah, and it really aligns with the goals of longevity research, not just extending lifespan, but actually extending health span, those healthy, vibrant years of life.
Yeah.
And speaking of health span, another study that caught my attention was published in Geroscience on March 7th. It's called Citrulline Regulates Macrophage Metabolism and Inflammation to Counter Aging in Mice.
Oh, the citrulline study, yeah. This one from the Shanghai Institute of Organic Chemistry is really interesting. It looks at the role of metabolism in aging, and
Yeah.
specifically the metabolism of macrophages.
Macrophages, those are the immune cells that go around gobbling up pathogens and cellular debris, right?
Mm.
What does citrulline have to do with that?
Yeah, that's right. So citrulline is an amino acid that's involved in a whole bunch of different metabolic processes. And what the study found was that citrulline levels actually decline with age in mice.
Okay, that's interesting. But why is that decline in citrulline significant? Like, what's the connection to aging?
Well, they found that when they supplemented citrulline in aged mice, it had some really remarkable effects. It reduced cellular senescence, protected against DNA damage, and it even improved some age-related phenotypes.
Wait, are you telling me that by simply giving these mice citrulline, they were able to slow down some of the negative effects of aging?
Yeah, that's exactly what their findings suggest. And what's even more intriguing is that they discovered that citrulline actually specifically targets macrophages. It modulates their metabolism and reduces inflammation.
So like they're giving these macrophages a boost, helping them do their job better, cleaning up the cellular debris that builds up with age.
Exactly. It's a fascinating example of how metabolic interventions could potentially play a role in extending health span.
It really makes you wonder what other metabolic tweaks we could make to slow down the aging process. Could there be a whole new class of longevity drugs out there that target metabolism?
That's a really interesting question. It's definitely an area that's ripe for more exploration.
Now, while we're on the topic of metabolism and aging, there's a paper published in NPJ Aging on March 6th that I want to bring up. It focuses on a fascinating creature called the spalax, also known as the blind mole rat.
Ah, the spalax. These little guys, these subterranean rodents, they're like practically the superheroes of the rodent world.
Mm.
They're known for their exceptional longevity and their resistance to cancer.
Exactly. And this paper from Johannes Gutenberg University Mainz takes a deep dive into the spalax's transcriptome, basically the complete set of RNA transcripts in its cells, to try to understand, how it's adapted to its unique environment and achieve this remarkable longevity.
Yeah, and they found some really interesting differences between the spalax and rats, particularly in genes that are related to cell death, antioxidant defense,
Right.
DNA repair, energy metabolism, immune response, even angiogenesis.
Wow, that's a lot. It sounds like the spalax has evolved this whole suite of genetic adaptations that contribute to its long lifespan and resilience.
Hmm.
Were there any specific findings that really stood out to you?
Well, one thing that jumped out at me was that in all the organs that they studied, which included the liver, kidney, and spleen, the Spalax had significantly higher levels of transcription of genes that are involved in genome stability maintenance and DNA repair compared to rats.
So they're better at keeping their DNA intact and repairing any damage that does occur.
Yeah.
Which makes sense, right, for a long-lived species.
Exactly. And another really interesting finding was that they also had lower expression of genes that are involved in aerobic energy metabolism and inflammation.
So it's like they've optimized their system for longevity by slowing down their metabolism and keeping inflammation in check.
You got it. It's like evolution has fine-tuned the Spalax's biology for survival and longevity in its unique environment.
And by studying these extraordinary creatures, we might be able to get some clues about potential strategies for enhancing human longevity. It's
Hmm.
like nature's offering us a blueprint for healthy aging, you know.
Absolutely.
Speaking of blueprints for healthy aging, there's a paper published on BioArchive on March 6th that I found really intriguing. It's from Newcastle University, and it focuses on a protein called PRDX6, a peroxyredoxin that's involved in regulating oxidative stress.
Ah, peroxyredoxins. These are fascinating proteins. They act as antioxidants, protecting cells from damage that's caused by those nasty reactive oxygen species, or
Yeah,
ROS.
they're like little cellular superheroes fighting off the damage that can lead to aging and disease. What did this study find about PRDX6?
Well, they actually studied PRDX6 in the nematode worm C. elegans, which is a popular model organism for aging research. And they found something really unexpected. The worms that lacked PRDX6 actually had an increased resistance to oxidative stress and lived longer under certain conditions.
Wait a minute, that doesn't make sense. I mean, we usually think of antioxidants as being good for longevity.
Yeah,
How could losing an antioxidant actually make them live longer?
it is counterintuitive, right? But here's the thing. They actually discovered that the absence of PRDX6 led to increased expression of another protein called FMO2, which is a flavin monoxygenase.
Okay, another protein of the long. So what does this FMO2 do?
So FMO2 actually has a pro-survival function. What that means is it protects cells from various types of stress, including oxidative stress.
So it's like losing PRDX6 triggered this sort of compensatory mechanism, a backup system that actually ended up being even more beneficial for the worms.
That's exactly what it seems like. And they found that FMO2 not only protected against oxidative stress, but it also protected against infections by bacteria and fungi.
Wow. So even though losing an antioxidant seemed like a bad thing,
Hmm.
it actually led to the activation of a more powerful protective mechanism. That's pretty amazing. It really shows you how complex and adaptable our bodies are.
It does, and it highlights the fact that there's still so much we just don't understand about the aging process. Sometimes inhibiting one pathway can actually have these unexpected benefits by activating other more beneficial pathways.
It's a good reminder to stay curious and keep exploring all possibilities when it comes to extending health span and lifespan. Now, speaking of exploring possibilities, there's a study published in Nature Chemistry on March 13th that really caught my attention. It dives into the world of biomolecular condensates.
Ah, yes. Biomolecular condensates. These are fascinating. These membraneless organelles that form inside cells are almost like these tiny little droplets.
Yeah, they've always kind of seemed a bit mysterious to me. What do these biomolecular condensates actually do, and why are they important for aging research?
So they're actually involved in a whole bunch of different cellular processes that are really crucial. Things like gene expression, protein synthesis, and signal transduction. They're kind of like little hubs of activity within the cell, but without any membranes to define them.
That's pretty cool. So how does aging affect these biomolecular condensates?
Well, this study, which is from Washington University in St. Louis, actually found that the electrochemical environments of these condensates are regulated by aging-associated intermolecular interactions.
Okay, so as we age, the way that these condensates interact with their surroundings changes. What are the implications of that?
Well, what they found was that these changes can actually affect the physical and chemical activities of the condensates, which can then disrupt cellular processes that are essential for maintaining health and longevity.
Oh, wow. So it's another example of how aging can affect our cells at a really fundamental level,
Yeah.
of affecting the function of these tiny little organelles within our cells.
Exactly. It's a reminder that aging is this multifaceted process that involves changes at various levels of biological organization. And understanding these changes is key if we want to develop, effective interventions to slow down or even reverse the aging process.
Now let's shift gears a bit and talk about some of the social and behavioral factors that influence aging. There's a fascinating study published in Brain on March 12th that really delves into the interplay of genetic and environmental factors in brain aging.
This study, which is from Zhejiang University, is really interesting because it used data from the UK Biobank, which is this massive biomedical database that contains, genetic and health information from hundreds of thousands of people.
It's amazing how much information we can get from these large-scale databases. What did this particular study reveal about brain aging?
So they used deep learning models to actually estimate brain age based on MRI scans, and then they conducted a genome-wide association study to identify genetic variants that are linked to differences in brain age.
So they were looking for connections between specific genes and how quickly the brain ages. Did they find any connections?
Yeah, they actually did. They identified 2 previously unreported genetic loci and 7 previously reported loci that were associated with brain aging.
Okay, but genes are only part of the story, right? What about like environmental and lifestyle factors? I mean, how do those play into brain aging?
Right. So to get a more complete picture, they integrated this genetic data with information about gene expression and protein levels, and they were able to prioritize 7 druggable genes that could potentially be targeted to slow down brain aging.
Okay, druggable genes. Now we're talking. Identifying specific genes that we can target with drugs to slow down or even reverse the aging process. I
Yeah.
mean, that's a major goal of longevity research.
It is. And what's even more promising is that some of these genes are actually already being targeted by existing drugs for other conditions. So this opens up the possibility of repurposing those drugs for anti-aging therapies, which could be huge.
You know, repurposing existing drugs, that's a brilliant strategy. It could really accelerate the development of anti-aging therapies. And this study provides like a really valuable roadmap for future research, highlighting these specific genes and pathways that are worth investigating further.
Absolutely. It's a great example of how, large-scale data analysis can really guide us towards some promising new avenues for intervention.
Now, speaking of roadmaps, there's another paper published on March 11th in Neuroscience that offers some really interesting insights into the modifiable risk factors for accelerated aging.
Ah, yes. This study from Zhejiang University School of Medicine used deep neural networks, or DNNs, to construct a biological age model based on data from the China Kadoorie Biobank.
And if I'm remembering correctly, they were actually able to use this model to identify certain factors that are associated with people aging faster or slower than expected.
Yeah, that's exactly right. They found that certain sociodemographic and lifestyle factors were strongly associated with accelerated aging.
Okay, so what are these factors that can actually speed up our biological clocks? I'm all ears.
Well, on the sociodemographic side, they found that being female, having lower education attainment, being widowed or divorced, and having lower household income were all associated with accelerated aging.
So it seems like social and economic factors can actually have a real impact on how quickly we age.
It really does highlight the fact that aging isn't just a biological process. It's also influenced by the context of our lives.
Yeah, it makes you realize that we need to be thinking about these social determinants of health if we want to create a society where everyone has the opportunity to age well.
I totally agree.
Now, what about lifestyle factors? Did the study find any modifiable behaviors that were linked to accelerated aging?
Yeah, they found that things like being underweight, not getting enough physical activity, and having poor sleep quality were all associated with aging faster than expected.
So it's the classic advice we've all heard a million times, maintain a healthy weight, stay active, get enough sleep. But now we're actually seeing how those lifestyle choices can really impact our biological age.
Yeah, it really underscores the power we have to actually influence our own aging process.
It's like we're not just passive bystanders in this whole aging thing. We can actually make choices that support our health and potentially slow down the aging process.
I think that's a really empowering message.
Now, speaking of choices that promote healthy aging, there's a fascinating paper in Aging, published on March 7th that I wanted to bring up. It's titled Gait Changes with Aging, an early warning sign for underlying pathology. It's from researchers at Radboud University Medical Center.
Gait changes, you mean like the way people walk? What does that have to do with longevity?
I know, right? It seems like such a simple thing, but this study suggests the subtle changes in gait, the kinds of things that we often just dismiss as just part of getting older, can actually be early indicators of underlying age-related diseases.
Oh, wow. So those seemingly minor stumbles, or shuffles could actually be a sign that something more serious is going on.
Exactly. Walking involves this really complex interaction between the nervous system, the muscles, and the bones, right?
Yeah.
So changes in any of those systems, whether it's due to things like Parkinson's disease, stroke, or even just normal age-related decline, they can show up in altered gait patterns.
So paying attention to these gait changes could actually lead to earlier diagnosis and treatment of these conditions, potentially preventing, more severe disability later on.
Exactly. It's like having a window into our overall health and a potential early warning system for age-related decline. And it reminds us to pay attention to those subtle changes in our bodies. They might be trying to tell us something important.
That's a good point.
Now, shifting gears a bit, there's a study on bioarchive published on March 6th from Seattle Children's Research Institute that takes a really deep dive into the microvasculature of the brain. The title is Impaired Capillary Venous Drainage Contributes to Gliosis and Demyelination in White Matter During Aging.
Oh, this is an interesting one. I remember reading this paper. They're really looking at, how the brain's blood supply changes with age, which is really important because as we get older, our brains become more vulnerable to damage from things like reduced blood flow and inflammation.
Right. And I know white matter is really crucial for communication between different parts of the brain. But what's this about impaired drainage? What's going on there?
So what they found was that using these really sophisticated imaging techniques in mice, that specific types of venules, which are the vessels that carry blood away from the brain tissue, they actually become constricted and less numerous with age.
So the brain isn't getting rid of waste products and fluids as efficiently as it used to.
Right, and that can lead to a buildup of toxins and inflammation in the white matter, which ultimately contributes to things like gliosis and demyelination.
Right. And gliosis is basically the formation of scar tissue in the brain, and demyelination is the loss of that protective myelin sheath around nerve fibers, which disrupts nerve impulses.
Exactly. So both of those processes can really disrupt the brain's communication networks and contribute to, cognitive decline.
So this study is really highlighting a specific vascular mechanism that drives age-related brain changes. Could this lead to new therapeutic targets?
Potentially, yes. If we could find ways to improve that capillary venous drainage in the aging brain, we might be able to slow down or even prevent some of these damaging processes.
Yeah, it's another reminder that maintaining a healthy circulatory system is absolutely crucial for healthy aging, not just for the body, but for the brain as well.
Absolutely. Now, one final paper that I think deserves attention is from the University of Virginia. It was published on March 10th on bioRxiv, and it's titled Diffusion MRI Microstructure Markers of Changes in the Human Brain Across the Lifespan.
Okay, diffusion MRI again. This seems to be a really hot topic in brain aging research.
Right.
What did this particular study look at?
Well, this study really delves into the use of diffusion MRI to actually track age-related brain changes in humans. And they use data from the Nathan Klein Institute's Rockland cohort, which includes brain MRI data from people of a wide range of ages, so they were able to create a really detailed map of how the brain's microstructure actually changes throughout life.
So like a roadmap of the aging brain. That's incredible.
Yeah, it is. And what they found was that certain measurements called signal fraction measurements, which basically reflect the movement of water molecules in the brain, they actually change in very predictable ways with age.
Okay, water molecules, brain scans. It's all starting to sound a little bit technical for me. Can you explain what those signal fraction measurements actually tell us about the aging brain in a little bit simpler terms?
Sure. Think of it like this. As we age, the spaces between our brain cells tend to increase, right? While the movement of water within those cells tends to decrease. So these signal fraction measurements actually capture those changes, and it gives us a glimpse into the brain's cellular environment as it ages.
So it's like they're taking these snapshots of what's happening at a microscopic level as the brain gets older. That's really fascinating. But why is that important? Like what can we actually do with this information?
Well, those snapshots can actually serve as a baseline for comparison when we're studying age-related brain diseases. So by understanding how the healthy brain ages, we can better identify deviations from those normal patterns that might actually signal a problem.
That makes a lot of sense. It's like having a reference point to see if things are going off track. Could
Yeah.
this lead to earlier diagnosis of brain diseases?
Exactly. And the earlier we can detect these deviations, the better our chances of intervening and maybe slowing down or even halting the progression of diseases like, Alzheimer's or Parkinson's.
That's a game changer. Well, we've covered so much ground today, from predicting telomere length with AI to slowing aging with plant extracts and creating these cutting-edge models for studying age-related diseases.
Hmm.
It's clear that the field of longevity research is just exploding with new discoveries and possibilities.
It really is. And what's so exciting is that we're not just talking about adding years to life, but actually adding life to years.
Exactly. It's about extending health span, not just lifespan.
It's about living longer, healthier, and more fulfilling lives.
And that's a goal worth striving for.
Absolutely.
Well, thank you so much for joining us on this deep dive into the latest longevity research. It's been such a pleasure exploring these breakthroughs with you.
It's been my pleasure as well. Always a great time discussing this fascinating field with you.
And to everyone listening, thank you for tuning in.
Mhm.
Until next time, keep diving deep.